am Dayang Nur Wahida binti Abang Zainuddin, Group 2, Subgroup 3, and matrix number 97354.
Hi, I'm Clarence and I'm from Group 2, Subgroup 3.
Hi, my name is Ahmad Tayyib Azamuddin Berkhalid and my matric number is 97219.
Hello, my name is Frankie Zheng Xiaohao. I'm from Group 2, Subgroup 3.
In this experiment, we focus on the unique characteristic of lipids, particularly their solubility in non-polar solvent and insolubility in water. Lipids are vital biological molecules involved in energy storage, cell membrane structure and cellular signaling. We examine different types of lipids such as fatty acid, triglyceride and phospholipid. Highlight their hydrophobic nature. Their physical properties like appearance and odor are influenced by molecular structure, including chain length and saturation. Lastly, we test lipids containing food products like olive oil, ghee, and shortening, showing how lipids contribute to flavor, texture, and nutrition in everyday food while demonstrating their solubility behavior. In this experiment, two sets of microcentrifuge tubes were prepared using 3 lipid-based food samples: olive oil, ghee, and shortening. A small amount of each sample was placed into separate tubes. The physical properties such as appearance and odor were observed and recorded. For the first set, water was added to test solubility in a polar solvent. The tubes were kept, shaken, and observations were noted. For the second set, toluene was added to examine solubility in non-polar solvent. Again, the sample were shaken and observed. All results were recorded in a table for analysis and comparison.
Firstly, 2 sets of 3 separate microcentrifuge tubes were prepared and 5 drops of all lipid-containing products were added into each tube. In set 1, 1 ml of water was added into the tubes. In set 2, 0.5 of toluene was added. Lastly, all the tubes are mixed well and observation was recorded.
In practical 3, we learn lipids and their physical characteristics. In this practical, we have tested the appearance, odor, and the solubility of 3 lipid-containing food samples. These samples were mixed separately with water and with toluene, a non-polar solvent. Through this, we discovered that lipids are insoluble in water but soluble in non-polar solvent, confirming their hydrophobic nature. And they have a unique physical traits that can be used for identification in food and biological systems. Additionally, this experiment helped reinforce the concept that lipids, while diverse in structure, are grouped based on their shared solubility behavior. We also explore real-life food labels to understand the types of fat they contain, such as saturated, unsaturated, and trans fat.
For experiment 3, which focused on physical properties of different lipids and fatty acids, handling oily samples was messy and sometimes led to sample loss. Achieving clear phase separation during solubility tests was tricky, especially if samples were not mixed completely. In addition, when adding non-polar solvents such as toluene, we need to conduct it in a fume hood and wear masks as a safety measure.